Hello everyone, myself Deepa Singh, working as assistant professor in Gandhar School of Pharmacy, SGVU Jaipur. Today we discuss about BCS classification, means biological classification system, and application of reformulation. And in this topic, mainly we include BCS classification. What is BCS classification? There's different types, there are classes, including examples. And this topic is the first unit topic, pre-formulation, and subject of industrial pharmacy 1, fifth sem. Now start this topic. Before starting this topic, you should know about this basic objects. In this objective parts, first you know about what is BCS classification, example of BCS classification, next definition or description of BCS classification. Next is classification of BCS classes, how you classify according to their different types, application of pre-formulation, what is the part of pre-formulation. In this, you know about application of pre-formulation, why pre-formulation is performed, and why it is important this pre-formulation study, and which field or which industry use this pre-formulation study. Next is importance of pre-formulation, why you perform this pre-formulation and the importance. So in this first we start by pharmaceutical classification system. In BCS classification system, the oral route of a drug administration is the route of choice for the formulator and continue to dominate the area of drug delivery technology. Means oral route is more appropriate and more good because everyone take by this route, and it show don't require any trained person or maybe any another person. You can take yourself by this oral route, and this is more appropriate to till now. And according to formulation wise, this route is also good for formulation purposes also, and mainly preference of this route is oral route. And in this oral route, there's an area of drug delivery is also large, and all this the popular this route is not free from limitation of absorption and viability in the villi of jet drug, but it is a certain problem, or maybe viability is also dependent on certain type of bacteria or GI tract absorption rate, and this is a limitation for absorption purpose. And the drug in the dosage form is released and dissolved in the surrounding GI fluid to form the solution. Means after taking the drug get released or dissolved in GI fluid or surrounding the GI and form the solution. Means it get disintegrate or dissolved, and after dissolution it is converted into solution form, and this solution form which is in a GI fluid or contact with GI fluid, and this is a rate limited step. Maybe it effect is observed by that particular type of the drug, and process of solubility is limited because not all type of drug or all drug solubilized in GI tract. So this is also solubility limited step. And in next part, once the drug in the solution form, it passes across the membrane of the cell, lining to GI tract, and the process is permeability limited. Once if the drug is passes to GI or by this process, it can easily cross this membrane or this cell lining of GI tract. So there may be chances that they do not show good permeability. Means if it's easily solubilized, then it may show certain permeability problem. So in this route, you can check both permeability as well as solubility problem, or check all both parameters are okay or not. If the drug is properly solubilized, so there may be chances that it do not permeate to that particular membrane or show permeability issue. If it's more permeability but it have less solubility, so you check both parameters, and onward the drug is absorbed in the systemic circulation. In short, the oral absorption and hence the viability of drug is determined by extent of drug solubility and permeability. Hence, this BCS classification or viability is mainly used to determine the solubility and permeability of rate of the drug, or how much drug is permeate or solubilized to that particular site. And the BCS is a scientific framework from classifying the drug substance based on their active solubility as related to dose and integration permeability. This is mainly depend on type of work or which drug is used, maybe active soluble or oil soluble. So you don't say that all your drug is pure impermeable or pure insoluble, but this solubility and permeability depend also on a type of the drug, maybe a hydrophilic in nature or maybe a lipophilic in nature. So according to this type of the drug, you decide which type of drug it is. and this type of properties of the maybe good and permeable or maybe good and soluble. If it's not in proper way, then you can modify by using this BCS classification, and with the help of BCS classification, you change the property maybe permeability, probability, or maybe solubility, permeability, both, and these BCS classification is based on only these two parameters, permeability and solubility. Next, when combined with dissolution of the drug product, the BCS takes into account three major factors that bonds or rate extent of the drug, absorption from IR solid oral dosage form, and IR is immediate release oral solid dosage form, dissolution, solubility, integration, permeability. Means that when you include BCS classification or your check drug, and after taking a drug, this amount or extent of the drug release or absorption of that particular drug is also depend on the type of dosage form. Suppose there is an example, IR solid dosage form. In this, you check on different parameters, three parameters like dissolution rate, how drug is dissolved or how it can dissolve in particular fluid medium, and solubility, how it easily solubilize, and interstitial permeability. And after this, how can it take place permeability rate, or how interstitial permeability takes place. Means not only amount of the drug, but type of the formulation is also vary. According to different type of formulation, you check this three parameter: dissolution, solubility, and permeability of the drug. And in this, you say that basic object of BCS classification is to predict in due performance of the drug product, and from in vitro measurement of permeability and solubility. Means you can check both in vivo and in vitro test. Not performing in vivo test, but in this you can say that when you're performing this test or by performing in vitro test, you can reduce this in vivo test, because before performing test on animal or before performing in vivo test, you just finalize your formulation good or not, or performing this in vitro test or with the help of this species classification. If it's showing problem, then you can maybe change your formulation or then show certain methods, and using of this animal is also reduced, because we don't directly perform the test on animals. We first perform in vitro test, and with the help of this in vitro test data, we move further proceed for in vivo test. Next, in this, improve the efficacy of drug development and review process by recommending a strategy identifying expenditure clinical BP test by equivalence test. In this, you can also improve the efficacy of the drug, or maybe improving the efficacy of drug, and you maybe enhance the bioavailability or bioequivalence of the drug. And to recommend method classification according to dosage form, resolution along with solubility, permeability characteristic of the product. And after performing this test, or after perform solubility, solubility, permeability, and the dissolution rate, you should know about that particular dosage form is good or not, a complete information of this parameter of that particular formulation. And so after this evaluating, recommended your formulation is good. If it's a good, then you say that this is okay, and you can perform the in vivo test. Next is to provide regulatory tool for replacing certain bioequivalence study by accurate in vitro dissolution test, or you can also use certain regulatory test or regulatory requirement by performing this in vitro test or bioequivalence study. With the help of this regulatory requirement, replace or maybe avoid in vivo test, and with the help of this in vitro test, you just finalize your formulation. And this will reduce the cost in the drug development process, also reduce unnecessary drug exposure in healthy objects. And so with the help of certain in vitro test, or with the help of this species classification, you also reduce cost, or maybe easily perform the test without expensive instrument, or without using this too much cost on that particular experiment. So this method is also very cheap, and do not require more cost, and you can perform this on healthy object also. And when you finalize your formulation, it's good for in vitro test. Until you just study BCS classification wise, or maybe according to in vitro test, provide guidance for industry. This is also important for industry purpose. Next is biological classification system and the types of BCS classification, and this is based on the solubility and permeability. The following biopharmaceutical classification system, that is BCS, recommended in the literature, Amidon 1995. Means this BCS classification is mainly divided into four class or four cases. That is, class one is high solubility and high permeability. Means in class one, your drug should be have high solubility and high permeability. Both are high. And in a case two, low solubility and high permeability, one is low, one is high. In this, solubility is less, but permeability is high. And in third part, there is high solubility and low permeability. Means one is high and the second is low. It's just opposite to the second one. In this, solubility high, permeability is low, and case 4 is low solubility and low permeability. Both cases are low, means solubility as well as permeability are low. And this is all about type of BCS classification, different classes of BCS classification according to depend on solubility and permeability rate of the drug. In this example of this class, first class is both solubility and permeability is high, and example is propanolol. And BCS type 2 class, solubility is low and permeability is high. Example is clamide. And third class is solubility high and permeability is low. And example is atenolol. And fourth class or last class, that is low solubility and permeability, both is low. Example is acetazolamide. This is the example according to BCS classification according to their solubility and permeability property. Next, in class 1, means in high solubility and high permeability. At class 1, the exit a high absorption number and high dissolution number. The rate limiting step is drug dissolution, and dissolution is very rapid than gastric emptying. And rate becomes a rate determining step, and rate of absorption is higher than the rate of excretion. Example: metoprolol, diazepam, verapamil, and propanol. This is the example of first class, and all this about drugs show high solubility and high permeability. And in this, you can say that class 1 is exhibit high absorption as well as high dissolution number, because it show high solubility as well as high permeability. Due to this, there is a high absorption and high dissolution also takes place, and this is the rate limiting step or limiting step for dissolution. If dissolution is not very rapid or gastric emptying is also rate determining step, or this gastric emptying affect your formulation. So in this condition, maybe it's a rate limited. Otherwise, it show more absorption and high dissolution rate due to the good solubility and permeability property. And the rate of excretion is also high because the low drug easily solubilized and fast formate. And due to this fast formation, excretion is also fast. Next, class 2. In this class 2, have a high absorption number but low dissolution number. Means absorption is more but dissolution takes place less. This is mainly, you can say that you formulate this type of drug on a controlled base, because less absorption takes place or slow absorption takes place, and but dissolution is fast. So you can modify the dissolution rate according to a requirement. And in vitro dissolution is then rate limiting step for absorption except a very high dose concentration. And this is in this class 2. In vivo drug dissolution or is also rate limited, or maybe depend on the certain condition, and because high dose is required for more absorption, and absorption is easily takes place by this class 2. The absorption for class 2 drug is usually slower than class 1. Because in class 1, high solubility and high permeability takes place. That's why more absorption is observed in class 1. But in this one, is solubility is low, then that's why absorption of class 2 is less. But it's a high, but compared to class 1, it is usually slow. Means more absorption is observed in class 1 compared to class 2, and occur a long period of a time, and it's so more absorption for a longer period of a time. And in vitro and in vivo correlation, that is IP-IBC is usually expected for class 1 and class 2 drug. This IP-IBC correlation is mainly expected or mainly depend on this class 1 and class 2 type of drug. And in this class 2 type drug, there is example: phenytoin, danazole, ketoconazole, mefenic acid, and nifedipine. This is an example of class 2. And next, third class is for the class 3 drug, permeability is rate limiting and the step for drug absorption. And these drugs exhibit a high variation in the rate and extent of the drug absorption. In this, there is also a certain permeability issue, or permeability is rate limited and the solubility is high. And the drug exhibit high variation in rate extent of the drug for absorption. And this also, there is a certain problem or exhibit a high variation or rate of extent absorption is also high. And since the dissolution is rapid and variation is attributed to alteration of physiological membrane permeability. Due to this high permeability, there's a dissolution is also fast or maybe show more permeability or more crossing through this barrier or membrane. In this example is cimetidine, acyclovir, neomycin, B and captopril. This is the example of class 3 drug. In class 4, drug exhibit a lot of problem for effective oral administration. Means there is so many problems due to low solubility as well as low permeability. Class 4 have both are in a low both solubility and permeability. So this class is rarely used because it shows so many problems or there's so many modifications required, and this will modify both solubility as well as permeability class also. And fortunately, extreme example of class 4 compound are exception rather than rule and are rarely developed reach the market. Nevertheless, the number of class 4 do the exist, and like Taxol and Crucifilin are the example of 4th class drug, which have low solubility and low permeability. Otherwise, these classes we are used with the more modification is required, low solubility and permeability rate, and very rare drug are come in market in class 4 drug because it shows more problem or more difficulty to solubilize or absorption rate. But there's no option for this class, and you still use this class, and you can change your formulation or modify your formulation by using this 4th class. In this classification can be used a basis for setting with dissolution specification, and can be also provide a basis for predicting the likelihood and achieving a successful in vivo and in vivo correlation. Means this basis classification is also used to IVIC correlation or by achieving good IVIC correlation in between correlation, and with the help of this BCS classification you can also correlate this IVIC correlation. And the solubility of a drug is determined by solving the highest net dose of the drug, that is 250 ml buffer adjusted between pH 1 to 8, and you can say that BCS classification performed by the particular required pH condition. Mainly pH condition is 1 to 8 pH, and volume taken of buffer is 250 ml. And drug substance is considered highly soluble when drug low solubility volume of solution is less than or equal to 250 ml. In this, you calculate how much drug is dissolved into 50 ml or how much volume is solubilized into 50 ml. And high permeability of drug is generally those with extended absorption and greater than 90% of the absence of its effect, or maybe showing viability. When suppose your drug showing 90% effect or maybe 90% absorption, then you can say that your drug is good. Documented instability of GI tract close to a permeability has been determined experimentally, and BCA suggests that high solubility and high permeability in case 1 drug, and some instant for high solubility and no permeability in case 3 drug. This case 1 and case 3 are more good because in this case 1 there is high solubility as well as high permeability, but in case 3 there is high solubility but low permeability. This type of class is been used for control release of a sustained based formulation due to low permeability but solubility is high, and you control your solubility rate according to the requirement. And in this, there's 85% is observed, and the dissolution in 0.1 normal HCl in 15 minutes. In this, mainly class 1 and class 3 drug, we use 0.1 normal HCl, and 15 minutes is required to perform this pathway study. And this is also rate limited for dissolution. In case of rate limited step for absorption gastric emptying, and this rate limited is depend on the gastric emptying time. Next is the mean P50% gastric resistance as emptying time is 50 to 20 minutes under a fasting condition. In this, almost 50% of time is required for emptying takes place within 15 to 20 minutes during fasting condition. If this information is conversion to conclusion is that drug product undergo 85% dissolution in 15 minutes under a mild condition. Test condition in 0.1 normal behave like solution and generally should not have any viability problem. Means within this 15 to 20 minutes, almost 80% dissolution takes place. And in this, you mainly use 0.1 normal HCl, and it should have good viability and no any problem is observed. It's an okay. If it do not show a good viability, then you can modify your formulation. If the dissolution is lower in gastric emptying, the dissolution profile with multiple time points multimedia is recommended. In case of low solubility or high permeability drug, case 2 type drug, having a high permeability and low solubility. The dissolution may be rate limiting step for absorption, and IVIVC may be expected. In class 2, there is IVIVC is also expected, means in vitro and vivo relation is also expected for class 2 drug in low solubility and high permeability, and it is also drug absorption or rate limited for absorption. And a resolution profile is a multimedia recommended drug product in the category, but in this for resolution study, multimedia solution is required and rate limited for absorption. In case of high solubility and high permeability, uh, high solubility and low permeability, that is class 3 drug, permeability is a rate limited step. Means in class 2, absorption is rate limited step, but in class 3, due to high solubility and low permeability is rate limiting step, and IVIVC will be possible in class 3 depending on the relative rates of dissolution, interstitial transit, and drug in case 4, that is low solubility and no permeability, there is a significant problem for oral drug delivery. Hence, this class 4 is rarely used. It's more problem or more error for oral drug delivery because it's both low solubility as well as permeability. Next is the oral formulation, mainly immediate use type of formulation, or maybe used in vitro test or animal study by predicting in vitro test. If this in vitro test is good, then you perform in vivo test, or cost is also reduced, or suffering of animals is also reduced by this BCS classification system. And this is mainly depend on solubility, permeability, and dissolution rate. And the certain factor that affect BCS is first solubility, and this maximum amount of solute dissolved in given solvent under standard condition of temperature, pressure, and pH. And the solubility is ability of drug to solution after dissolution. Means in this you can say that solubility is mainly depend on how much drug is dissolved or solubilize in a particular solution and solvent in particular solvent. And according to this solubility is depend on the conditions like which temperature or how much pressure is applied or pH condition. Hence you calculate how easily it gets dissolved given temperature, pressure, and pH condition. And in this you calculate ability of that particular drug to solubilize or dissolve in a solution. And highest single dose is completely soluble in 250 ml at pH 1 to 6.8 at 37 degrees Celsius. It's an example. If you are taking a single dose, then it should be completely solubilized in 250 ml solution, or pH condition is 1 to 6.8 according to your formulation that where the effect is required, and it should be between 1 to 6.8, and buffer solution in 250 ml, and it's completely solubilized at body temperature, that is 37 degrees Celsius. Next is permeability. In permeability of the drug to pass the biological membrane, which is lipophilic, and this is permeability is mainly depend on lipophilicity of the drug, and permeability is indirectly based on the extent of absorption of the drug absorption, means how much drug extent is available or indirectly available for the absorption, and it is also depend on the nature of the drug and lipophilicity or hydrophilicity. Drug substance is considered to be highly permeable when extent of absorption is determined by 90% or 90% or more than administered comparing to reference dose. This permeability calculate how much drug is permeable. It should be up to 90% or more than 90%, and it is up to 90%. Then you use in vivo test to finalize your formulation. Otherwise, maybe certain changes required or you can modify the formulation. Next is dissolution. In dissolution, it is a process in which solid substance solubilized in a given solvent and mass transfer solid surface to liquid phase. Means that dissolution is the process in which solid substance solubilized in a given solvent, or maybe from how much drug solubilized in a particular media and dissolve in a particular media. And this dissolution test mainly performed with the help of USP apparatus 1 at 100 rpm or USP apparatus 2 at 50 rpm. Means you can use any one of this USP apparatus. So apparatus type 1 or 2. If you are using one, then the speed of requirement is 100 rpm, and if you are using apparatus 2, then the speed required is 50 rpm. And in the solution media, 900 ml in 0.1 normal FCl or stimulated gastric fluid, that is pH 1.2, and point 0.1 normal HCl solution is required, and pH is 1.2 without enzyme, and pH buffer 4.5 and pH 6.8 buffer is also required if you are taking multiple media and stimulated interfacial food without enzyme. Means this dissolution test is depending on the type of formulation. If you require effect on the stomach, then in this condition you require 0.1 normal HCl, and pH is 1.5 without enzyme because effect is required on stomach. If you're preparing formulation for interstitial effect, in this condition pH is 4.5 is mainly preferred, and you can also prefer 6.8 buffer, and you perform this test according to interstitial fluid without enzyme, and you perform this dissolution test with the help of different USP apparatus, and mainly we used USP 1 and 2 apparatus for this dissolution test, and speed also maintain, and temperature is mainly body temperature 37 degrees Celsius is required. But if you are using apparatus 1, 100 rpm speed is that, and in case of apparatus 2, 50 rpm is required, and you feel this buffer solution up to 900 ml in the solution apparatus withdraw, and withdraw every interval of time, 10 minutes or maybe half an hour. And after withdrawing this sample, you should maintain the same condition. Since how much sample you withdraw, you should add that amount of that much amount of the sample in your solution. Why pre-formulation is important, and importance of pre-formulation. In this pre-formulation, we describe the process of optimizing the delivery of a drug through determination of physical chemical properties of the new drug molecule, and that affect the drug performance and development of a stable and safe dosage form. Means with the help of this pre-formulation, you can formulate or manufacture suitable physical and chemical property of your drug, or maybe before starting formulation or formulating your drug, you just formalize your formulation is okay, and show a good physical and chemical property, and it do not show any performance issue, and it should form stable and safe dosage form. Means overall it is a good and ready to formulation, and you just assure all things are okay. Next is pre-formulation study for new drug molecule provide useful information for subsequent formulation of physicochemical stable and biopharmaceutical. In this, you can say that this study is also helpful drug molecule or provide useful information for subsequent formation of physicochemical stable and biopharmaceutical drug. And the next is preliminary evaluation. In this, once pharmacologically active compound has been identified, different project team has been responsible for ensuring the compound enter in the development process and optimum molecule form. If your formulation should have a good pre-formulation study and show good result, then you can finalize your formulation or enter your drug for in vivo test or pharmacological active compound identification, and this is mainly performed with the help of certain animal or with the help of certain other technique, and you finalize optimize your molecular form. And if your deficiency is detected, then project team decide on molecular modification. If any problem is occurred during preliminary study or any deficiency is detected, then a certain expertise or team decide which modification is required, or after modification this problem is overcome or not. If not overcome, then change your formulation. Otherwise, you may modify drug or may modify the excipient. Example: salts, product, solvent, and polymer. These are the modification. If your drug is not suitable for that particular form, then you change maybe in the salt form, product form, or solvent form, or maybe polymer form. And mostly salt and product approaches are common. This salt and product approach are mostly common, and it shows similarity. And there's a little bit difference between the salt and product. Otherwise, almost same. Most of organic compounds are formed by addition or removal of water. Salt are mainly formed due to the organic compound or by the addition and removal of proton from ionized molecule, which is then neutralized with counter ion. And after this, it is neutralized with counter ion. An example is epinephrine hydrochloride. Organic salt are more stable soluble than unionized molecule. Organic salts are more good and more stable and more water soluble than unionized molecule. And the prodrug are synthetic derivative of drug molecule. In synthetic derivation or synthetic changes of a drug is known as prodrug, and it is a more pure form, and just change your formulation on your drug to another one or more appropriate or more pure form. It is known as prodrug, and it's a synthetic form or synthetic derivative of that particular drug. And salt is more water soluble and more appropriate, more solubilized in water compared to an ionized molecule. And pharmaceutical improvements from product include stabilization, increase or decrease solubility. In this, you can say that pharmaceutical improvement is improved stabilization or increase or decrease of solubility, crystallinity, taste, odor. Means in this, you consider solubility and crystallinity, taste, odor. All these parameters are depend on product. If you change into product and means change your drug into product, and there are certain properties also change, then the chances that it improves or will increase or decrease the solubility, crystallinity, as well as test on other stability problems also. An example of this is erythromycin isolate. Once the optimal molecular form of the drug has been selected, formulation development begins. There's a task in drug development process. After optimization, there's a formulation start or maybe begin. And in this application of pre-formulation, introduction to drug discovery and role of pre-formulation. Means first in this application part of pre-formulation, which field uses pre-formulation. Introduction of a drug discovery and role of pre-formulation means you discover certain drug or synthesize certain type of drug, and you start formulating. Just assure that your drug is good for coming in the market or overall property is good. So introduction of a drug discovery and role of formulation study, use pre-formulation, introduction to drug development and role of pre-formulation. Before starting drug development or how you perform this pre-formulation study, you should know about this pre-formulation parameter or role of your drug. Next is component of a profiling, fundamental development of delivery, and next is solubility and permeability study. Next is stability, means this pre-formulation is not limited for certain type of drug discovery, but also you can check solubility and permeability problem, which type of a drug is solubilized or permeable too easily, and different type of drug property, fundamental property. Next is solid state property, which state is more suitable or more appropriate for your formulation of a particular drug, even solid form or liquid form. Next is pre-formulation report, and in this you can also perform pre-formulation report, or with the help of this pre-formulation report you proceed further step. And pre-formulation study for genetic product and generic product. Suppose you are just putting your formulation or preparing generic product. You know difference between generic and branded product. Branded means newly synthesized formulation and having a patent for 15 to 20 years. And after this patent, you copy this product with the allowance or acceptance of that particular branded synthesized person. And during this manufacturing or formulation of generic product, you just perform this pre-formulation study or in vivo test. There's no requirement for in vivo test. You just perform in vitro test, because already this drug is compile all this information and come in the market. So in generic product, we also perform this pre-formulation study. Next, in this application part, you should know about what is a pre-formulation. This is all about pre-formulation and BCS classification. And after completion of this topic, you know about complete information about BCS classification. What is BCS classification? How you check this BCS classification? Means first to explain or classify, or maybe explain detail what is BCS and the example according to this 4 classes of this BCS. To give certain example, which example is suitable for class 1, 2, 3, and 4. And according to this, you can know about how you perform this stability study, and which is a very limited step, maybe solubility, permeability, and dissolution. And you also know about and this BCS classification is mainly depend on 3 parameters: solubility, permeability, and dissolution rate. And according to this 3 parameter, you can select which type of class of a drug. And application of the pre-formulation. Why pre-formulation is important, and which field use this pre-formulation for genetic preparation, or you use the animal study, or with the help of in vivo study, you just finalize your formulation and start performing in vivo test. If certain changes is required, then you can modify or change your formulation during pre-formulation study. Means overall pre-formulation is very important for your formulation or for your drug manufacturing for particular dosage form. And BCS classification is also important because this gives the information regarding your drug, which type of drug is which class of the drug. So according to this type of the drug, you can changes your drug or maybe select a class 1, 2, 3, and 4. According to the requirement, you modify your formulation. This is all about learning outcomes. You should know complete information about these topics after this PPT. Next is reference part. In reference part, there are different books are available for this subject, and some books which are having a more good matter and content are this. In this first is Ansel Pharmaceutical Dosage Form and Drug Delivery System, 8th edition by Lloyd B. Allen, Nicholas G. Popovich, Howard C. Ansel, published by BI Publication PVT Limited. Second reference is Pharmaceutical Pre-Formulation by JTC Cartesian, published by Technological Publishing Co. And third one is Textbook of Physical Pharmaceutical of CBSE Subramanian, published by Balla Prakashan. And fourth one is Theory of and Practice of Industrial Pharmacy by Leon and Lackman, Habers E. Lieberman, Joseph L. King, third. This is also good book. And this Leon and Lackman. Complete information of pre-formulation as well as this topic is covered in this book. And next is Martin Physical Pharmacy, Pharmaceutical Science, 5th edition. This is also good book, and in Martin all topics are covered with good content, and sufficient detailed information is given in Martin book. Next, sixth one is Pharmaceutical Dosage Form Tablet, Volume 1, edited by Happeton, Lieberman, Leon Dyckman, published by Marcel and Becker. This is all about the reference. These all books are good. You can use any one book or maybe a combination of this above book. Next is question part. In question part, first is a non-crystalline form that is often more soluble than a corresponding crystal form. In this, you know about pre-formulation study or what is crystal form. So according to this study, you should answer this question and select out of this 4 options anyone. In this, A option is dissolution, B is solubility, C is co-solvent, and D is amorphous. And the second question is the presence of a co-solvent can increase the solubility of hydrophobic organic chemical. In this, you justify that when you add co-solvent or presence of a co-solvent, how solubility gets affected or solubility of hydrophobic organic chemical gets changed. In this, A option is co-solvent effect, B option is dissolution, and C option is capillary melting method, and D is organoleptic property. And in third question, which of the following statement is false? Out of this ABCD option, you just justify which option is wrong or which option is false. And in this A option is a liquid formulation for intravenous delivery must be aqueous solution that do not precipitate in the bloodstream. Means liquid formulation that is given by intravenous or IV route, deliver in aqueous solution form and do not precipitate in your blood. It shows good property. And B option, parenteral suspension and oil-based parenteral solution can only be administered by intramuscular or subcutaneous route. Means parenteral suspension or oil-based parenteral. This suspension also may be in oil form, or oil-based solution you can administer by only by intramuscular route or by subcutaneous route. We do not use any other alternative route or any other parenteral route. And the C option is small volume parenteral liquid must be administered intravenously, but large volume parenteral liquid must be administered by any route. Means parenteral is divided into 2 parts, small and large volume parenterals. And in this point, we said that a small volume of parenteral have a limited amount or limited ability for drug administration. That is, you administer only by IV route, but large volume of parenteral you administer by any route, oral route or parenteral route or any other route. And in D question, what for injection is term used to describe the sterile purified water, and that is reserved for the preparation of aqueous medicine for parenteral delivery. Means used for water for injection, and this water for injection is required for sterile purified form and for preparation of aqueous medicine for parenteral delivery. And this is 4 option. In this, you select any one option which is a false. All 3 options are true. And out of this ABCD, you decide which one is false or which one is wrong. Next is in-process tests are done. This mainly in-process test is done for A is reduce the cost, or maybe reduce the amount, or reduce the economical purpose for in-process test, and to consumer acceptance, for acceptance of consumer or acceptance of that particular person consumer. C is to minimize the risk of product failure, also reduce the product failure risk or minimize the product failure risk. D option is none of the mentioned, not option in ABC. So you can select any one option. In process test done for what? And fifth question is non-pharmaceutical additive. The formulation ingredient must be highest what and lowest what. Non-pharmaceutical additive. In this, you select ABCD. What is a non-pharmaceutical additive? And the option you can select pharmaceutical ingredient must be have highest what and lowest what. B is option is what parental is the most common for all injectable product. Almost all parental are the common, or all are injected by parental product. And C option is a process in which you can produce product that loves the dry state. You may manufacture or process form a product that allows a dry state. And since large volume of fluid is administered, what must they contain? Large amount of fluid is administered by this parental route, or you may use pharmaceutical additives. So non-pharmaceutical additive use any one option ABCD, which is correct. Next, sixth question is the nature of preparation, that is ingredient. And in this option A is how does ionization radiation kill, and B is filtration sterilization resulting. In this first, you know about question, the nature and preparation and this ingredient. You know about how to prepare that particular ingredient or nature of preparation. And this A option is by the help of what does ionization radiation kill. Radiation helps to kill that particular bacteria. Base filtration sterilization result. That is also sterilization technique, means with the help of filtration sterilization. Says what are the formulation ingredient, what type of formulation ingredient is used in a formulation. Base what does sterilization method depend on, which method is depend on the sterilization. And next is seventh question, FTR assay and limit test are mandatory for testing of this FTI test. You know, this we already covered in pre-formulation, and in this you know about this FTI test is assay and limited test or mandatory for testing which type of product. A1 is finished product, B is raw material, C is in process material, and D is none of the mentioned, means not mentioned in ABCD. And you just select any one option, maybe finished product means final product, or maybe before starting a formulation, raw material or in process material, or not mentioned in that particular option ABC. Next, its question is why emulsion dosage form for lipophilic drug work better. In this A option is easy manufacturing, why emulsion are more good and more lipophilic drug. In this select out of this ABCD, A is easy manufacturing, B is the highest stability of the drug emulsion form, and C is prescribed a large volume surface area of oil to the GIT for absorption of the drug, and D is a fast clear from the plasma. Next, out of this ABCD option, you select which one is good for emulsion or lipophilic emulsion. It is easily manufactured, maybe high solubility or stability of the surface area, to GIT absorption of fast clear to the plasma. Select which one is appropriate or correct for this eighth question. Next, ninth question, which is the major rate limiting step in absorption of the drug or from suspension dosage form. In this, you select which type of formulation have a more absorption in suspension dosage form. First is tablet disintegration to granules, tablets converted into particles, or maybe converted into small particle or into granules. B is a granule disintegration to fine particle, maybe it get converted into granules into more fine particle, more small particle, then fine particle into dissolution, then converting fine particle to dissolution process, and dissolution absorbing the blood, then dissolution it converted into or goes into for absorption into the blood. So out of this ABCD option, you select any correct option, or which option is correct for absorption of the drug in suspension form. Maybe it converted into fine particle, or maybe converted into more small particle from granules, or maybe easily available for dissolution and dissolution to absorption. Out of this ABCD, to select any one correct option regarding this ninth question. The next last 10th question is all oral solid dosage forms show. First is greatest chemical and microbiological stability. Or oral solid dosage form. In this include maybe a solid or maybe liquid dosage form that you've taken by oral route. Means both solid and liquid you can include in this 10th question. And this is greatest chemical and microbiological stability. It should have means good chemical as well as microbiological stability, not contain foreign particle or bacteria, and chemical stability is also good. More impurity or oral solid dosage form also have more impurity in the option. C option is more administration problem. Oral route show more administration problem in C option. And D is more irritation. Means you can say when you take your drug from oral route, you show more irritation. So out of this A, B, C, D option, you select any one option which is correct for oral solid dosage form. And you know that in this question asking about all oral solid dosage form, like not solid dosage form but all oral dosage form. It may be not single solid dosage form or maybe not only single liquid dosage form. In this question asking about oral dosage form. Oral dosage form may be include both solid, liquid or any type of the formulation. So out of this A, B, C, D option, you select any one option for this 10th question. This is all regarding your question part or regarding your problem solving question. After completing this topic or completing this PPT, you should know about all this question answer on easily solve these questions and you clear your concept by the help of this question. So thank you so much. Thank you for listening and watching this video. Thank you so much, everyone.